Thank you, Brenda. So, emerging data, genome scale data, is exceeding our capacity to model it biologically. This is especially true in the case of disease association information, where we're constantly learning new things about not only how genetic variation is associated with human disease, but also how genes interact with other genes and how genes interact with environments. This is a lot to ask of mammalian models. So even though they offer incredibly faithful models of human disease, there's an incredible investment required for making them a single knockout mouse. So having methods to inform your experimental work before you begin are incredibly useful in terms of maximizing your experimental resources. So zebrafish are small, transparent, fecund, fast-growing. They develop similarly to higher vertebrates, but notably, there's an increasing number of methods for targeted gene disruption. So they're a good platform for kind of evaluating large-scale phenotype screening prioritization methods. So the goal of this project was simple. What we wanted to do was develop a machine learning framework where we predicted gene phenotype association on a large scale across multiple phenotypes. We were going to directly validate our predictions in zebrafish using morpholinos. For anyone that's not familiar, a morpholino is an oligonucleotide that you customize that can target any transcript in the genome. So what you do is you inject it at the one-cell developmental stage, and it lasts for about 5 days. So you basically subtract that transcript from the process of development. And the idea was for this to be iterative, that we could use our phenotype predictions to inform the next phase of machine learning, to make more accurate and more specific predictions. I'm not going to go into too much detail about the machine learning framework. It was based on the approach by Tian et al. It was a combination guilt by profiling, guilt by association. So in guilt by profiling, your goal is to predict gene phenotype associations. Our training information was 338 anatomical terms that are maintained by a zebrafish public database. And they also maintained morpholino phenotype association information. So we had about 700 morpholinos, and we knew how they affected each of these 338 anatomical processes. Gene features were things like expression information, protein domain information, phylogenetic profiles, gene ontology, and KEGG annotations. And there was a separate random forest classifier that was constructed for each phenotypic term, for each of the 338 anatomical terms. In guilt by association, you're predicting gene-gene associations. So training information was any genes that share a phenotype, and features were things like genetic and protein interactions mapped from other species, co-expression, similarity in phylogenetic profile. You make one random forest classifier that generates a functional linkage network, which is mined for every phenotype, and then you combine the scores through logistic regression. So when this was all done, what we had was a matrix, 15,000 zebrafish genes by 338 anatomical processes. And every element of that matrix was the likelihood that that gene would affect that anatomical process when you knock it down with a morpholino. So these data are recently impressed. It's not online yet, but you can search these using GeneMania or using the RothLab ZFunk website. But what I'm trying to show you here is that this is precision through cross-validation. And we do better for some phenotypic terms than others. We tended to do very well for cardiovascular and neuronal phenotypes. But even among cardiovascular phenotypes, which are shown in panel B, there's a lot of variation. And what we think this variation is due to is the consistency with which these phenotypes have been observed. So this is all based on community annotation. Some phenotypes are very easy to observe consistently, and some phenotypes not so much. So we picked one phenotype that was a general term describing cardiac function, had a 72% precision at 20% recall. That's in panel C. Which wasn't our best performing term, and it wasn't our worst performing term, but this is the one we chose for follow-up. And just looking at the genes that were prioritized, the ones that are highlighted in yellow had external evidence of having a cardiac phenotype. So they had mutant information, or they had morpholino information that had been uploaded to ZFIN since we obtained the data. So this is good independent confirmation that we're on the right track with our predictions. But a lot of these genes still had unknown functions, so we decided to follow them up. And when I say follow them up, what did we do? I injected morpholinos targeting these genes into zebrafish, and I took videos of the beating hearts at 48 hours post-fertilization. This is a 48-hour embryo. This is a control embryo. It's a two-chamber heart. The atrium beats, then the ventricle. You can see the blood flowing through normally. This is a completely morphologically normal heart. Okay, see if you can spot the difference. This is a playing video. And this highlights something interesting about zebrafish physiology. They get enough oxygen through diffusion in their first few days of life that they don't need a circulatory system, They don't need it to function necessarily. So this fish will swim along completely normally until it drops dead at about 5 days post-fertilization. This was a gene that had been labeled as being a skeletal isoform of troponin, based on its similarity to human troponin, but clearly has a cardiac function in zebrafish. Of course, not all the phenotypes are this plainly observable. So what I did was I came up with a semi-quantitative scoring scheme. I looked at 5 parameters of cardiac function, and I gave each a score between 1 and 4, where 1 is closest to wild type, and 4 is the most severe defect. I took videos of each of these morpholinos at 48 hours post-fertilization. I randomized the names of the files so I didn't know what I was evaluating, and this is a delta score. So it's a measure of how much worse are you than your controls from your particular day. I looked at 16 test genes. For 12 of these genes, there was a significant defect in at least one of these 5 parameters, as compared to 0 of 5 negative control genes. The negative control genes were genes that we had ranked lowly with our prioritization scheme. When I followed these up with more quantitative assays, 11 of these 12 had measurable defects in valvular function, ventricular function, electrophysiological development, calcium handling. I'm not going to discuss those in detail, but I am going to talk about one, which was TMEM88A. So this is a gene that had no known cardiac function, but had known to be involved in Wnt signaling. We heard a little bit about Wnt signaling this morning, but just briefly, Wnt signaling is pervasive among all vertebrates. It's got a lot of developmental functions, but one of its functions is cell-cell communication. And previous work by my lab has shown that Wnt is involved in development of electrophysiological coupling in the ventricle. So this is an isolated zebrafish heart, and we've stained it with a transmembrane potential sensitive dye. And through high-speed photography, you can track the action potential as it propagates through the zebrafish heart. And in past research, we've shown that there's the conduction velocity of this wave is faster at the bottom, the outer curvature of the ventricle down here, than it is at the inner curvature. This is a natural property of vertebrate heart development, and this is due to Wnt signaling. So when I look at TMEM88 knockdown, so these are isochronal maps basically showing you in a static image what I just showed you in the video. This is a wild-type heart, and these are 2 hearts that have had TMEM88A knocked down through two different morpholinos. There's actually an increase in cell coupling. So this kind of cell coupling effect is exacerbated. I looked at expression of connexins, which are a cell coupling protein, and cell architecture. There were no other differences that could explain this increase in electrophysiological coupling. So it was a Wnt-dependent effect. So at this point of the talk, I hope that I've shown you that you can generate phenotype predictions at scale. They're reasonably good across all phenotypes where you have good training information, but I've only shown you one potential treatment. I've only talked about morpholinos. So how translatable is this to other treatments? So in separate work, what I did was I took 4,182 known yeast bioactive compounds, and I screened them in 96 well plates in zebrafish. And so what I did was I just basically looked at 48 hours post-fertilization, and I wrote down all the phenotypes that I saw in each well. And certain phenotypes came out as being prevalent: cardiac disorders, pigmentation defects, developmental delay, body axis defects. So for the 6 most prominent phenotypes, again, I created learners. And since we're looking at compounds here, excuse me, compounds and not genes, I used chemical properties. So I looked at molecular fingerprints in terms of secondary structures, molecular weight, total polar surface area, other molecular properties. We did, again, better for some phenotypes than others. Notably, we did poorly for a phenotype called cranial edema, which it was a passenger phenotype. So cranial edema was something I never saw on its own. It only occurred with other phenotypes. And this, again, speaks to you need rigorous and consistent training information to have a good learner. But through both area under the curve and precision recall, I think we did pretty well for these 6 predictors. Well enough that I prioritized the 900,000 compounds that are available from Cambridge. And these ranked lists seem to be consistent with the phenotypes that I was expecting. So for example, if I prioritize these 900,000 compounds using the learner that had been trained to predict cardiac phenotypes, I was preferentially upregulating analogs of known vasodilators. If I did this for body axis phenotypes, I was upregulating compounds that are antineoplastics, which are useful as chemotherapeutics. I rescreened the top 10 compounds for each of these 6 phenotypes, and for 5 out of the 6, I saw a significant enrichment in phenotypes that I expected over the bioactive set. The one that I missed was cranial edema, and I already talked about why that might be. And this is just a diagram to show you that these compound predictions are structurally diverse. So these are all of the compounds that have a known or predicted cardiac phenotype. The ones in white are the ones from my screen that I actually observed a cardiac phenotype for. The ones in blue, yellow, and red are predictions. So blue are ones that I haven't tested yet. Yellow are ones that I've tested and saw no phenotype for, and red are ones that I saw a phenotype for. And of course, this is highly significant. So 11 of my top 12 ranked compounds had an instantly recognizable cardiac phenotype and no other phenotypes. But something that I wanted to highlight with this network diagram, so the edges between the nodes are structural similarity among compounds. And it's not just, we're not just finding analogs of compounds that we screened. It's a combination of molecular features and structural features. So if you look at these orphans at the bottom, these ones that have no high structural similarity to any other compounds, we're still significantly enriched for the phenotype that we expect. So it's kind of akin to maybe the Lipinski rules in combination with structural features. So to kind of summarize where we're at with this project, I think that vertebrate phenotypes are predictable if you have sufficient training information. I think that this is useful across a range of phenotypes. Obviously, we focused experimentally on cardiovascular phenotypes, but this wasn't our necessarily best performing predictor. I think you could use this for anything. And I don't see this as being restricted to zebrafish. There's a paltry amount of phenotypic information and structural and experimental information for zebrafish compared to, let's say, mouse, rat, or yeast. So this is what I see as a minimum bound. I see this as these phenotypes tend to improve, as the data improves, these predictions can only increase in value and increase in precision. One of the focuses of our lab is to establish an automated screening platform for phenotypes. As we get this information online, these predictions are going to, I think, increase substantially. And so with that, I'd like to thank the members of the McCrae Lab, Fritz Roth and the members of his lab as well, Quaid Morris for their help with Gene Mania, and the members of the Zahn Lab. Thank you.
Questions for Dave? In that last diagram you showed with the compounds, were you just showing us data for the predictions for the cardiac?
Yes.
Yes. Okay.
Yeah, so it did range a little bit. So if I had less training information, it did seem like it was a little bit more dependent on structure, but that's the one I had the most information for, so that's what I used. Tim.
So Gabe, one of the things in the, there was this last summer a paper from the group at the Sanger where they systematically looked at mouse knockouts, and one of their conclusions was that they get new phenotypes out of previously characterized genes just as often as anything else.
Yeah.
I was wondering if you could see that either in your analyses or just in the predictions.
Yeah, so that's a fantastic question. I mean, it's not something I've looked at specifically, but this is the value of this. Is if you build this data into the model, there's no reason why you can't predict environmental specific functions of genes. And this is something I think you need a phenotype prediction method to inform your experiments with, because this is unpredictable, right? And you can't possibly assay all experimental conditions for a given knockout and be expected to see the phenotypes.
Well, but I think what the mouse groups have been proposing, I mean, is that they actually, that's an important thing to do, is not to just screen for one thing on all the morpholinos, but to what they seem to be doing with the mouse mutants, maybe just because it costs so much to make a mouse, is, you know, like Toronto, we have this whole building, which is the Toronto Phenotyping Center, or whatever it's called, for precisely that reason.
But I mean, so there's effectively no end to the environments that you could elicit, right? Like, so there's not only, I mean, this is especially difficult in mouse, and it's not necessarily as observable as zebrafish. You can't see as many phenotypes with the naked eye. You'd have to be looking for them specifically. So even if you look at all these environmental conditions, the matrix has got to be giant. And so I'm sure you could do a bit better than if you were just looking under laboratory conditions, but that's a huge undertaking.
So it seems that the whole phenotyping is based on morphology and things you can just detect by just looking at the animal.
Yeah.
What about molecular phenotyping? What about
that's a
RNA-seq and all these things? Because to me, for example, that is a vast deficiency of, let's say, mouse knockout programs, that they focus on 30, 50, 100, 200 phenotypes when there's a potential of millions. But if you do a molecular profiling, you can actually represent all those if you actually analyze the data in an appropriate way. So have you done any of this, or are you planning to do? How does it relate if you have?
So that's a great question, and I think it relates also to what Tim was saying as well. So this, I mean, there's no end to the number of phenotypes you can measure. And when you start to build in molecular data and specific, like you start to consider gene expression as a phenotype, you need some type of prediction method to get at this. I don't see any reason why it couldn't work. It's not something I've worked on specifically. I've only focused on more of the morphological effects, but it's a fantastic area to begin to work in.
Okay, thanks a lot.
Thank you.
We should move on.